Good morning, everybody. Welcome to Pediatric Grand Rounds. Just a reminder for those of you who are joining us remotely on WebEx, you can feel free to type your questions in the chat at any time, and I will ask them to our speaker at the end of the lecture time. I'm going to bring up Dr. Mark Wainwright to introduce our lectureship and our speaker today.
Welcome to the 2023 annual AtrophySarkowsky Lecture in Child Neurology. It's my honor to introduce Dr. Richard Finkel to give this presentation. I'd like first to acknowledge the support of the Sarkowsky Foundation for making this lectureship possible. This first began in 1995, and also for the Sarkowsky Foundation and the family support for the Division of Pediatric Neurology and its academic activities now for many years. Herman and Faye Sarkowsky were married for 66 years until Herman's death in 2014. He was a businessman in Seattle and at one time co-owner and founder of the Portland Trailblazers and the Seattle Seahawks. This foundation was established in 1982 and had and continues to have a focus on Jewish culture, healthcare, pediatric healthcare, and the arts. Their involvement in the division began in the early 1990s with the endowment of the chair that I'm honored to hold, as well as an endowment which made possible this lectureship, and it continues to this day with recent gifts enabling us to continue clinical research in child neurology. So this foundation has had a powerful effect, a long-standing effect on the academic activities of the Division of Pediatric Neurology at Children's and the University of Washington. This particular gift, this lectureship, was established to allow us to establish a lectureship which would invite people who had made fundamental and important contributions to the field of child neurology, broadly defined. And we're honored today in exactly that spirit to have Dr. Finkel giving this lectureship. The lectures have been given since 1995 by luminaries in our field. I'll just mention a few, which will be familiar to the neurologist on the call. Jean Accardi, Charlotte Gervais, Karen Nelson, recently Nina Shaw. There are many others. This will give you an idea of the stature of the people who've given this lecture. Dr. Finkel is currently the Director of Neurotherapeutics at St. Jude's Research Center. He is a neuromuscular neurologist. His training was at Washington University, St. Louis, and Boston Children's. He practiced and led clinical programs and research programs in neuromuscular neurology at Children's Colorado, Nemours, Children's Hospital of Philadelphia, and since 2020, his position at St. Jude. Dr. Finkel's published nearly 250 academic papers. He's received numerous rewards, all in the field of neuromuscular disorders in children, inherited neuropathies, Duchenne's muscular dystrophy, and spinal muscular atrophy, which will be the focus of his talk today. The volume of his contributions does not do justice to the impact that he's had. He's led as a PI over 30 trials for the treatment of these disorders, resulting in the development of 7 drugs for their treatment, including the landmark work in the development of nusinersen and many other drugs, which have transformed the practice of pediatric neuromuscular medicine. This is really now at the cutting edge of the synthesis between the diagnosis of genetic disorders and their treatment. He has led some of the seminal work in this field, and it's our honor to have him here today to give this presentation.
Thank you, Mark, for that lovely introduction. And indeed, it's a pleasure to be here and speak with you today. I hope that AtrophySarkowsky is participating remotely, or other members of the foundation, and I want to acknowledge their generous support to the hospital and to the neurology department here, and to make this lectureship possible. Today, I'm going to share with you a personal journey that goes back over 20 years. And we're going to use the example of spinal muscular atrophy, or SMA, as what I think is a paradigm that could serve well for the treatment of gene-targeted diseases for numerous other genetically based conditions, both in children and adults. These are my disclosures. And to be compliant with CME, I do want to mention that I will be discussing some off-label and investigational use of 3 drugs today, so stay tuned. So our objectives here is I'm going to first give a brief overview of what do we mean by rare and ultra-rare diseases and why is SMA a good example of that, and then try to use that to further talk about the discovery, innovation, disruption, and transformation in the evaluation and treatment of these patients with spinal muscular atrophy. And then to step back a little bit and just give one example where I've dipped my toe in the water with a prenatal treatment of an infant and sort of highlight some of the particular opportunities and challenges therein. So rare diseases are defined by the FDA, it's in legislation, of a prevalence of less than 200,000 individuals. So it's not all that rare, and that's in the US. And there's a different definition in Europe, but for the US, and that's important because drug development and a lot of things get special favors from the FDA if you're trying to promote a drug for a rare disease, as defined here. But there's also this group called ultra rare, loosely defined as less than 7,000 individuals, or 1 in 20 million in the United States. And if you use that definition of rare disease, there are about 7,000 different rare diseases, an average of 4,000 per disease. And some may have a single individual or 5 or 10, and others may be up to 200,000, but the mean is about 4,000. And that's not really viable for a drug company to develop a drug for at least 1,000, maybe 4,000, but 1,000 is probably the cut point of where it becomes viable. So there's going to be this whole realm of patients that have ultra-rare diseases that are being largely neglected. They're orphan diseases. We know what they have. We know their gene. We know they're targetable, but we may not be in a position to generate a drug for them. So there are efforts going on to try to address that. And currently, if you do the math, about 1 in 10 individuals in the United States has a rare disease. 2/3 of these are children. About 30% die. These are catastrophic diseases, die before reaching their 5th birthday. And of these, about 80% are genetically based. And unfortunately, only 350 of these 7,000, about 5%, have an FDA-approved drug. And very few of those are actually gene-targeted. A lot of those are working on downstream modifier pathways. They do some good, but they're not really hitting the target as such. So when we talk about spinal muscular atrophy, I put in parentheses was because it was the most common fatal disease of infancy until these new treatments came along. The incidence is about 1 in 11,000 live births. And interestingly, that's changing a bit now that parents are having carrier screening. And so that's a little bit of a wobbly number. The prevalence was about 20,000, and it's actually come down. Maybe it's 15,000 now. And it's largely a pediatric disease. Importantly, there's a high carrier frequency of about 1 in 40, and there is some ethnic variability here, as low as 1 in 32, or high in sub-Saharan Africa, 1 in 70. So typically in a room such as this, there's going to be one person who's a carrier of SMA. And the American Board of OBGYN does recommend that all women who are thinking of becoming pregnant or in their first trimester of pregnancy be tested for SMA on the panel. And then if you test positive, then your partner should be tested. And that way you have an idea whether the fetus is affected, because this is an autosomal recessive disease, 25% chance of recurrence with each pregnancy. Okay, so this busy slide highlights the history of spinal muscular atrophy, starting from the beginning of the description by Verdnick and Hoffman in the late 19th century. And the first century was really to define the disease pathologically and clinically, as shown in the top here. Now, the bottom row is busy, and that starts in 1995 with the discovery of the causative gene for spinal muscular atrophy. It's called survival motor neuron, or SMN gene, by Judith Melke and her colleagues in Paris. And after that, it set off a whirlwind of activity, both in the laboratory to develop transgenic animal models, but also a rash of drug discovery efforts. And I'm going to highlight some of those. But also importantly, the NIH targeted SMA for special funding. And this was unique in their experience. Usually you would go through the standard R01 kind of funding mechanism. Here they said, we think there's really a treatment opportunity here based on the molecular genetics. So they had a special set of grants to support both preclinical and clinical drug development for this disease. So I'm going to highlight that in the next 10 years, there was a lot of work in the laboratory to try to develop antisense oligonucleotides, to develop a gene therapy, and also small molecule drugs for the treatment of SMA. And you can see that the first of these, nusinersen, that Dr. Wainwright mentioned, was approved by the FDA in 2016, followed by a gene therapy in 2019, and then a small molecule drug called risdiplam in 2020. So we now have 3 FDA-approved drugs, and I'm going to go through each of those and some of the issues and the learning involved. So here's the standard neuropathology. We think of SMA as a motor neuron disease. It affects the nerve cells in the ventral horn of the spinal cord. They send out their long axons that innervate skeletal muscle. And that's true of the nerve cells in the cranial nerves, the motor cranial nerves that feed the bulbar muscles as well. So you see the very thin ventral roots, and there's a paucity of motor neurons in the ventral horn. But SMA actually is beyond that. There's also changes pathologically in the dorsal root ganglia. So that's the sensory system, also in motor cortex and even thalamus. So now that we're treating drugs that are focusing on the motor neuron, are we going to unmask some other areas of pathology? Because our drugs aren't getting to motor cortex or to thalamus. And are these related to some of the early learning issues that we're starting to focus on, some of the other issues in SMA? So there's still a lot to be learned. And Charlotte Sumner at Hopkins has been particularly persuasive to say we still need to get autopsies on these patients. There's particularly the treated patients. Does the drug get to where it's supposed to? Is it doing what it's supposed to? Is it increasing the deficient protein, SMN protein? When you think of SMA, you think, oh, this is a neurodegenerative disease. But in fact, there's also a neurodevelopmental arrest that's a component. And shown on the left here is a fetal muscle biopsy from some 50 years ago nearly, from Marina Husmanova in Poland. And she showed that all of these little tiny muscle cells are really fetal muscle and if you looked at a type 1 infant, it looks the same. It's almost like there was a failure of maturation of the fetal muscle. Whereas in the older patient with more advanced disease, you get what's called grouped atrophy, which is typical of diseases like ALS or SMA. So very different. So the important thing that we're trying to figure out still is what's this developmental arrest all about? And when do we need to treat to try to promote optimal response to these wonderful drugs? So when you think about a motor neuron, you think about the cell body that lives in the spinal cord. It sends out its long axon, and then there's the neuromuscular junction. And shown here is an abnormal neuromuscular junction on the left as compared to a normal control. And so what we're learning is some of the early pathology, including what you could test in the EMG lab, is reflected at the neuromuscular junction. So some of the fatigue almost has a myasthenic feel to it. So we're starting to target where in the motor neuron is exactly the pathology, the clinical features coming from. There's abundant work being done now to try to better understand fatigue at a physiological level. So the disease manifestations, if you read the book, it'll say these babies are normal at birth. Well, that's not always right, and I'll show you why. It's characterized by weakness, hypotonia, areflexia, proximal more than distal, legs more than arms, also involving the neck and trunk, bulbar muscles, tongue fasciculation, respiratory muscles with the diaphragm relatively spared, and the paradoxical breathing pattern, which I'll show you in a moment in a video. And all of that continues to be true, but now that we're treating these babies, we're seeing different phenotypes emerging. So it's not like you're delaying the same phenotype. We're seeing a whole different set of characteristics in these babies. There are also secondary musculoskeletal deformities. And you'll read that cognition is intact and that there are smart verbal communicative, and that's true largely, but in the type 1, the most severe babies, maybe not so much. So we're starting to learn with more detailed interrogation that there may in fact be some pockets of cognitive developmental delay. What about sensation? I showed you before that the dorsal ganglia can be affected. So we're now paying more attention to say, is the drug getting to that, or are we starting to see some sensory deficits over time? So this is the typical characterization that goes back to the 1980s. Type 1, the most severe form, by this little baby here, presents under 6 months of age. It represents more than half of all of SMA have this type 1. And they never sit, they never roll, and few reach their second birthday, even with supportive care. Type 2 is called the intermediate form, or Dubowitz disease, presents between 6 and 18 months of age and reflects about a quarter of patients. And these children do achieve independent sitting, as shown in the little boy here, who cannot raise his hands up any higher than that. But they never walk, by definition. And they have typically a normal lifespan. Particularly now, it's improving well into adulthood. And type 3 are only about 1 in 7 who present after 18 months into the adult years, and they achieve walking, but unfortunately about half of those children will lose walking by their 10th birthday, and eventually most of them lose ambulation. So that's the spectrum, and we artificially broke it down into these groups, types 1, 2, 3. We're actually getting away from that because it doesn't really fit anymore the patients. Now we're putting them into functional categories: sitters, non-sitters, sitters, walkers, and they transition because they don't fit this traditional natural history phenotype. So I just want to highlight on this video, the baby on the left is a normal twin, Christopher, and you can see I'm lifting him up and he's having a good old time. And on the right is his twin sister, Charlotte, who's, you'll see, she's not very happy. So I'm lifting up Christopher and he's just holding his head up, just nice, neck extension, and you'll see. And then watch Charlotte. I'm just going to lift her up and she's going to slide right through my hands in a moment, right about now. And I'm just trying to steady her and see she's just struggling, and there's no weight-bearing effort, as was with Christopher. And she's getting fussy, so we're going to stop very soon. But I just want to turn her over into prone, and she has the omega sign, the inverted, and she's not lifting her head back, and no neck extension, no weight-bearing effort. So Charlotte was born before any of these drugs were available, and she received supportive care, and she died 2 months later of a respiratory illness, which was unfortunately the typical scenario. So it was palliative care, supportive care, genetic counseling, and deciding, well, with G-tubes and BiPAP and all that, were we really doing much? We were extending life, but we weren't really increasing quality of life. It was a very difficult time for those of us treating these babies. And if you did give them maximal care, this is what it often looked like. So this is an end-stage patient who's 32 months, different patient, trach, G-tube. She's awake, and I'm asking her to move her hands, and if you look really carefully, you'll see just a hint of finger movement. And she can blink her eyes to communicate, but boy, that's a really hard sight to see. Unfortunately, we don't see this anymore. That's gone away, so that's a textbook video. So let's go to the genetics of SMA. It's a monogenic disease. It's autosomal recessive. It's called a loss of function disorder because it's a deficiency of a protein called SMN protein. Okay, and there are 2 genes. There's the SMN1 gene on chromosome 5, shown here, and then there's a paralog gene. It's not quite identical, and it's very near it. It's not adjacent exactly, but it's very near it, also in 5q. Okay, and it has one important difference, one nucleotide at position 840. So where a C should be, it's a T. Okay, and what's really curious is that C to T change does not change the amino acid. It changes the splicing efficiency, so that exon 7, that's highlighted by this little arrow right here, this exon 7 is often excluded from the transcript. Not always, sometimes it's included, but most of the time it's excluded because it's a wobbly splice site because of this variant. Okay. So that gene makes the SMN protein. So here's our SMN1 and our SMN2 gene, and SMN1 makes abundant protein. SMN2, only about 10% of the amount of protein that's generated from the SMN1 gene. And 90%, because of that splice site being wobbly and failing to include exon 7, it makes an unstable protein that's degraded. Now, the number of copies of that SMN2 gene is very predictive of the phenotype. Not 100%, but the more copies you have of that backup SMN2 gene, the milder the disease phenotype, as shown here. So here's, if you have 2 copies of SMN2, you're likely to be a type 1. If you have 3 copies, you're likely to be a type 2, the sitter. But it's not 100%. So you could predict, if you get a genetic test report back and it says there's 2 copies of SMN2, you could say, well, you're likely to be type 1, but you could be, there's still a 20% chance you could be the milder type 2 phenotype. So you have to be really careful in learning how to interpret these genetic test reports. Now, the number of copies of SMN2 is related to survival. So if you have 1 copy, 2 copies, these are the Kaplan-Meier curves, 3 copies, or if you have 4 copies or more, you have a normal life expectancy. So this is improving life expectancy as you have increasing numbers of copies of SMN2. But let's look at it a little differently. And this is patients that have classic SMA type 1, Wernicke-Hoffmann disease. And if you look at the babies that have 2 copies of SMN2, they have a precipitous linear decline. And those with 3 copies, who are less frequently seen, a somewhat more indolent decline. So the clinical trials were set up on the basis of including patients that have 2 copies, because it was a more predictable phenotype. So the median survival was, if you have 2 copies, was 10.5 months, and only 8% reached a survival of 20 months, even with supportive care in this cohort study. There are gene modifiers that I'm not going to go into today. There are other biomarkers besides the copy number, and I will go into some of these further in the talk. So let's think about the strategies for how you're going to treat SMA. Okay, so here's a patient with SMA because you can see the SMA1 gene is missing. It's missing because there's no functional copy. It was deleted or mutated. It has a variant. So you're totally dependent on the backup SMN2 gene. So strategy 1 is replace the SMN1 gene. That's a gene replacement therapy, and that's one of the 3 drugs that's currently available for treatment of SMA, onasemagene and Epivec. The other 2 treatments that are currently available focus on the SMN2 gene. So what it's trying to do, think of this as a seesaw, is to try to change it from 90 and 10, change the splicing efficiency with this drug so it's more like 50-50. So if you get a higher inclusion of exon 7, then you're going to have more of this protein being made that you need, this very critical protein. So now I'm going to talk about nusinersen, the first of these drugs to get FDA approval. And this is an antisense oligonucleotide, and it modulates the splicing of the SMN2 gene to promote inclusion of exon 7. Now, this is given intrathecally, and it's been remarkably well tolerated, even in little babies, where you have to do repeated administrations of loading doses and then maintenance doses every 4 months. Surprisingly, these babies tolerate that very well. And the drug itself has a strong safety profile, and it's really shown to be spectacular. And one of the things that we learned is that the earlier after diagnosis, the better the response, which sort of makes sense. But the drug was sort of rushed to FDA approval based on an effective dose in 2016. There's now a study in process to say, well, can we even do better with a higher dose? Because they never did dose optimization. They took a dose that worked and said, let's get it out and see if we can help children, and then we'll fine tune it later. So that's what's going on now. Now, how does this drug work? Okay, so here's exon 7, here's exon 8, here's the intron, or the DNA that's in between exon 7 and 8. And there are these blocking factors called HNRNPs. Now, what nusinersen does is it comes in, and nusinersen is this red. It's a string of 18 nucleotides. That's what it is. But it was designed by Adrian Craner at Cold Spring Harbor and others to match by Watson-Crick base pairing exactly this one area in the genome. We think this is a unique sequence here. The ASO latches onto that. It displaces those other proteins. It allows this thing to bind, which is really important, called a U1 SNRP. And once that binds, then it promotes the inclusion of exon 7. It doesn't actually fix the original problem. That C to T change at 859, it's still there, but this is a sort of a backdoor way to promote inclusion, to get more exon 7 inclusion and to make more of the protein that you're missing. So it's a very elegant concept that came out of the lab. And then boom, you have exon 7 and now you make more protein. Okay, so I'm just going to highlight something which I was involved in, which to me was just beyond belief. And this was called a phase 2 study, a proof of concept study. We already sort of knew the drug worked in animals and in cell culture. Now we're trying it in children. Okay, type 1 children. And this was the natural history course that I showed you before. And this is showing improved survival of these babies on this Kaplan-Meier curve on babies treated. This was an open label study. So everyone got drug. And then we did 2 different motor measures, something called the Heine, something called the CHOP INTEND. And again, I guess full disclosure, this is a motor measure I co-developed when I was at CHOP. So that's why we called it the CHOP INTEND, with a really good physical therapist. But what we started to notice is that, gee, here we have a neurodegenerative disease where these kids get weaker day by day. And here we're seeing that they're actually getting stronger. And nobody expected to see that. We thought, oh, maybe we can slow down the rate of decline. Maybe we can stabilize it. But to see actual improvement in these very weak symptomatic babies was really an eye-opener. Okay, so here's a baby, Cameron, and these videos are all with the permission of the family, and he has his own YouTube site, which I'll show you in a minute. So here he is. He's a pretty symptomatic little guy. See his legs, frog leg posture, paradoxical breathing, paucity of movement, couldn't even keep his head straight. And here he is just a couple months later. He's holding his head up. Now you might say, oh, that's cute. No, they never do that. Once they've lost head control, and most of them never have it, to be able to hold your head up and maintain it, and I am not propping him up there. Now, here he is, and I'm going to lift him up. Remember Charlotte? So, but he can tip his head and bring it back up. You never saw that. So those were some very early signs. And here he is where he's pushing down against my hand. Again, he should be collapsing. There should be no resistive effort. And this is a home video. And we started to learn the parents can make a lot of observations that we're not doing in the clinic with our silly testing. So here I'm seeing not only can he do it, but he can maintain it. He can keep his arms up against gravity and reach for that toy for quite a while. So there's trying to look at fatigue and sustaining something. And this was a watch this, Dr. Finkel moment. Okay. Well, I told you before, they never roll over. So I had to jump to oh my God, he's going to roll off the table. So here he is, but it took him 14 months. As you know, babies are typically rolling 4 to 6 months. So delayed, but hey, this is amazing. Okay, another home video. Okay, there. Now this is something that we didn't capture in the clinic, but is very clinically meaningful today. He's sitting, now notice, he's sitting up without a hand prop, and he's got pretty good fine motor. Well, we didn't even think about looking at fine motor. Okay, now we are. So here's the most recent one. He is still really weak. He's still fed by G-tube. He's on BiPAP at night. He goes to school. So I love that. Okay. But they engage him in a soccer team. He's like their extra goalie. He does scuba diving with his dad. Who would have imagined? Okay. So after we had this first treatment in 2016, there were efforts made to say, okay, maybe we should start identifying these babies pre-symptomatically. So there were two efforts. One in 2006 to get SMA on the newborn screening panel, and it was turned down because they said, well, there's no treatment. You have to have a treatment. Otherwise, technically, there's no reason to identify them. So once there was the first treatment in 2016, we went back to Health and Human Services, which is an FDA panel, and they said, okay, yeah, now we can do it. And this sort of follows on the whole newborn screening, started by Dr. Guthrie for PKU. This is a DNA test, and there already was a DNA test for SCID. So this was really easy to do because you already were capturing DNA. Okay, you didn't have to draw more blood. You can do it from the blood spot. Okay, and you can get those results back on day 5 or day 7, along with all the other metabolic diseases that are looked for. Okay, so now dial forward. In the past 5 years since the feds did it, each state has to go and decide, are we going to add it to our panel at the state level, and are we going to fund it? So, and the answer is yes. So now 48 of the 50 states and 98% of babies in the United States are being screened for SMA. So symptomatic SMA, as I showed you in those earlier videos, is largely going to disappear because the patients who want to be treated are already being treated. Not all, but the majority are. And all the new patients, pretty much 95%, are going to be picked up really early. And presumably the parents are going to want treatment. This testing, however, does not pick up 5% of patients with a particular genotype because it only looks at the 95% that have a deletion on both alleles of SMN1. And 5% have a deletion on one allele and a point mutation on the other allele. And currently that's not being screened for. Now, if we get to widespread newborn whole genome sequencing, which I hear is coming, well, that might change. But for now, we still will see an occasional symptomatic patient crop up in the clinic because they weren't picked up on the newborn screening. Okay. So on the left is a little boy whose sister died of traditional typical SMA at 7 months of age. And he was picked up because of the risk, the familial risk. And he was treated at 9 days of age with nusinersen. And you can see he's sitting, he had good head control. He's throwing the toy the way, children do. And we're starting to focus on his fine motor more. We're learning we need to do better. Okay, that's my fine motor test. And then here he's not happy at all. He's being a toddler. I don't want to be in a clinical trial. Okay, but now, so he's demonstrating that he's walking. Not only that, but he's walking within the time frame of a typically developing child. Okay, and then the little girl on the right was just too cute to not include. She's ready to go to work, and she has 3 copies, whereas Chance on the left has 2 copies. So does that make a difference? Well, yes, it makes a huge difference. So in this study, I want to highlight these upper 2 lines as compared to these. So the clinical trials that looked at symptomatic patients showed this increase in a test called it's a motor milestone test called the Hammersmith Infant Neurologic Exam. And these are the babies treated pre-symptomatically or shortly after birth with nusinersen. And these are the 2-copy babies here in the blue, and in the light blue are those with 3 copies. And you can see there's a divergence. There's a divergence. That the 3-copy babies do really well, and they actually hit a ceiling effect. It's not that there's a plateau. They can't do any better on this scale. So we had to transition them to a different scale. Whereas you can see the 2-copy babies are doing phenomenally well, but not quite as well as the 3-copy. So remember that, because I'll show you a little more why that's important. Okay, let's turn to the second treatment, onisimagene epipervivac. Okay, which is a single administration. You treat once. It's an IV administration of an adeno-associated 9 vector with, it includes the full SMN1 gene. So that is the cDNA. And it's a wampum dose of 1.1 e to the 14th. That's 10 to the 14th viral genomes, viral particles. So you're infecting a child with a, quote, non-infectious virus that's been gutted and replaced with this SMN1 gene, but you're given it all at once over an hour, intravenously. So of course there's going to be an immune response. So even though the virus, the AAV, is not a pathogenic virus, it can cause a wampum immune response. And this was approved in 2019, but only up to 2 years of age. Nusinersen was approved for all children, all ages, all types of SMA, types 1, 2, 3. FDA said, this is a big deal. We're going to only approve it up to age 2 years. In Europe, they have different labeling guidelines. It goes up to 21 kilos, independent of age. So try to figure that out. So it's very different. So there have been some remarkable results, and I'm just going to highlight a little bit here. But one of the important things is that even though the gene therapy is targeting motor neurons, the transduction, the infection, if you wish, of cells is 300 to 1,000-fold higher in liver cells than it is in motor neurons. And the toxicity is mainly hepatotoxicity and the risk of liver failure. So even though it's targeting one thing, it's a systemic administration. Once you've given it, you can't take it back out. You can't turn it off, as is true of other gene therapies. Having said that, the results are spectacular, and now over 3,000 children have been treated with this drug. This is what the gene therapy looks like. So this is a cartoon shared by Avexis, now part of Novartis. So here's the AAV9 virus, and it has this capsid shell. You've heard all about capsid proteins from our friend COVID. Okay, so this is the gene that's been put in. That is the SMN1 gene. And it has a promoter, and it has an enhancer, and it's a self-complementary. So it means normally AAV single-stranded. This was made to be double-stranded. So once you deliver it and it finds its way to the motor neuron nucleus, boom, it's ready to set up shop. And it doesn't technically integrate into the host genome. It stays in the nucleus as what's called an episome, as a little packet of DNA. Okay, and it's going to keep churning out SMN. And how sustainable is this? Well, so far it looks really good. These children are now up to 5 to 7 years of age, and we're not seeing the effect wear off. So far, so good. But let's return to how did the children with 2 copies do, who were identified on newborn screening and treated within the first 6 weeks of life with this drug, onisimagene epipervivac. So this highlighted here between these bars is the age for developing independent walking. I think, is this the walking one? So let's assume that's walking. I can't even read it. So this is the normal time window according to the, it's walking alone, unassisted, which is shown here at the bottom. Okay. Now, if you notice in green, all these dots are individual patients. They all achieved walking between these purple bars, so meaning within the normal time range expected for a typically developing child. Whereas if you notice those with 2 copies of SMN2, about half were between the bars and half are beyond. So they're doing spectacularly well, but we're learning that even treating them at 2, 3, 4 weeks of age may not be soon enough. And they may have already lost some motor neurons during fetal development. And in fact, there's fairly good pathologic data to support that. here's another way to look at it. This is the Bailey scale that you're familiar with, and this is looking at the gross motor scale. So here's the 3-copy babies, and this is a scale that aggregates all sorts of different factors, not just walking. And it looks at it by age over time. All the 3-copy babies are within the gray zone for typically developing children. For 2 copies, I've highlighted in blue here that about half, they're making gains, no doubt, but they're fallen below that lower limit. So they're lagging a little bit. They're not normal. So this is raising other questions. Would combination treatment be a good idea? If one drug is good, is 2 drugs better? Well, how do you define if these children have a real capacity to actually improve by the addition of a second drug? We don't know that yet. I want to turn to the third drug, risdiplam. This is a small molecule drug, and this is shown here. Small molecule has molecular weight of 401 Daltons. The dosing is age and weight based. It's per kilo. It's given orally once a day. It was FDA approved in 2000 for all ages and all types, similar to nusinersen, all ages, all types. The problem is the insurers don't always cover it for all ages and all types. They look at it more narrowly, but technically the FDA says everyone with SMA should have the opportunity to be treated with either nusinersen or risdiplam. And it is also an SMN2 splicing modifier, similar to what I showed you earlier. It, however, has a narrow therapeutic safety index, meaning you can't go too high in dose as you're going to start courting some toxicity potentially. Okay. And just like we saw with the other drugs, it works really well in an early onset, recently diagnosed symptomatic type 1, less so in type 2. Children who are older, more advanced, for some reason don't see, it's more of a stabilizing, slow improvement. It doesn't have that POW effect the way we see in the type 1s. And it also works spectacularly well in these pre-symptomatic neonates under 6 months of age. And I'm using pre-symptomatic in quotes because we're now getting the idea that about half of these babies really are symptomatic. It's very subtle, but they're not normal, particularly if you do some additional studies to characterize them. So with that, the opportunity came up for me to treat a patient last August. It actually came up about this time last year. So it was a woman and her husband who had a baby who died at 14 months of age with SMA and had 2 copies. And she's pregnant again and had an amnio, and the fetus is affected with SMA and has 2 copies of SMN2. And we know from epidemiologic studies that the combination of 2 copies and a positive family history of a baby with type 1 is 99+% predictive that this fetus is going to have the severe type 1 phenotype. And the parents were pretty smart. They already knew about the 3 drugs, and they called me, and I did a telemedicine second opinion consultation, which is something I do. And it was like, well, what else can you do for me today? Thank you. I'm not sure, but so I considered, well, is this the kind of case that we might consider treating the fetus before birth? Okay, and there was a strong rationale for this, and there has been fetal treatment before. So on the left is intrauterine transfusion for a form of thalassemia, alpha thalassemia. Well, that's not gene therapy. You're doing repeated transfusions. And then just a few months ago, there was a really interesting report from Tippy McKenzie's group at UCSF of in utero enzyme replacement therapy. So again, you're not targeting the gene for Pompe disease, glycogen storage disease, but rather you're providing the deficient gene product, in this case, the enzyme, alpha glucosidase. Okay, so here, my thought was, well, can we take it a step further and maybe give a drug that targets the gene? So I'm not going to go into all of this, but there was a strong rationale to this. We went and got a lot of scientific advice from Roche, and the people are listed there. to say, am I crazy to even think about it? Do you have enough preclinical data? Is this even safe to do? What have you learned in mice and rabbits and monkeys and all that to support this? And then I went to our ethics committee and said, am I crazy to even think about this? Tell me to go away, or, is this something that really is legitimate? And then we worked on a protocol development, and we reached out to the FDA, and there's a program officer there, Annie Wynn, who was particularly helpful in sort of guiding us, because this had not been done before. And the clock is ticking. Like, we can't take 6 months because the baby will be born. So at the end of the day, the FDA approved what's called a single patient investigational new drug. Our IRB approved it. The mom came to St. Jude in Memphis, and we did all sorts of baseline testing on her, and what we could indirectly do on the baby. And then we started treating the mom with the adult dose, the FDA approved dose, as if she had SMA. She's a carrier, but she doesn't have SMA. But we knew that the drug goes transplacentally, it crosses over and gets into the fetus, but we don't know how much. So we started at the adult dose, and then after birth, we drew cord blood levels, and it took us a week to get the drug level back because we didn't want to overshoot. And then once we got the level back, and as we had predicted, it was in the modest range, then we started the baby on drug on day of life 8. And she continues on this, and still is now. She's 7 months old. And this busy slide just goes through all the different things we did for the mom and the baby, and the mom's in white and the baby's in black. And we did weekly, ultrasounds and biophysical profiles, and drew blood on mom. And then we drew a whole bunch of blood, cord blood. And then after birth, we looked at the washout in the mom because she stopped taking the drug, and we looked at for the baby the increase in dose once she was started on the drug orally, and we got the adverse effects. And it's amazing how many adverse effects a pregnant woman has once you start characterizing it, but none of them seem to be related to drug. And then we did a lot of assessments on the fetus and on the baby. The baby did have some problems. There was a C-section. The baby had retained fetal fluid. A NICU doctor kind of freaked out because said, "Well, this baby has SMA." So they intubated her for about 6 hours and watched her in the NICU, and then extubated her, but because she had wet lungs, which we attributed to the C-section, not to the drug. And then since then, her breathing has been fine. And also, she was found to have a heart murmur, and then echo showed a VSD and a tiny one. And we had to go back and say, oh, well, VSDs occur in the second trimester, well before this fetus was exposed to drugs. So we don't think that's related. Now, there was a little problem after birth where the mom said, she's not a great feeder. I have to wake her up a little bit to feed her. And I was watching her weight gain, and you can see here that there was a little plateau. She lost the typical weight in the first week, but she didn't really start gaining weight. There was a plateau for 3 weeks. So I think that may be a drug-related side effect. I don't know, but so we had to watch it. But we didn't stop the drug. We continued, and she didn't have GERD, or she wasn't a spitty baby. There wasn't anything else. But since then, she's fed maybe too well. She's, I'm trying to temper that a little bit. Okay. And then we did a bunch of testing, and I'm not going to go through this for the sake of time other than to say her development's been normal as far as at 18 weeks when we did some sophisticated testing, including the Bailey, a feeding scale, electrophysiologic measures, muscle ultrasound. We did drug levels. We did the SMN protein, and we found that the baby's protein was very similar to that in a drug study of this same drug called the Firefish study. So it seemed to be falling in the range. It's only an N of 1, but looking pretty good. And then we looked at something called neurofilament levels. Neurofilament is a very non-specific biomarker. It goes up in virtually all neurologic diseases, MS, stroke, Alzheimer's, Parkinson's disease, anything with neurodegeneration, it goes up. But it also, in particular, goes up in neuromuscular diseases because it's derived largely from the axon. Okay, so it's a biomarker of axonal damage and decay. So we have the mom, and she's here, and there's two types. There's a neurofilament heavy chain and a light chain. We'll just focus on the yellow one here. And this was before delivery and then after delivery, maybe from her C-section stress, it went up a little bit and then it's coming down. But let's look at the baby. So the baby on the neurofilament heavy chain, and this is from cord blood, was 266. You say, oh, okay. But we know it's higher in all children, the first year of life, because the nervous system's maturing. So if you say, well, how does that compare to other things? Well, here's that one of the studies, the Nurture study, that was treated, these were babies treated with nusinersen, identified within the first 6 weeks of life. And they maybe weren't all pre-symptomatic, but they were picked up really early and treated, again, within the first 6 weeks. And using the same assay, the same machine, this done the same way, the baseline pre-treatment was nearly 21,000 versus 266. So lower is better. Higher means there's more axonal damage. So even though these babies were looking really good, there was something going on in the background. So we think collectively, oh, here she is. I just have to show you this. So here's our first rolling over. Come on. Yunk. Come on, you can do it. There she is. Okay, so she's looking pretty good. Now, we're not going to make any claims on an N of 1, and it's too early. I need to follow her. But this has created some interest to say, well, should we be looking at this more broadly? Because there are more babies being identified prenatally. So I'm going to just wrap up. We don't need to go into all this other than to say we basically showed that it's feasible and seems safe in this N of 1 to administer this drug, risdiplam, to a mother. There were no ill effects. There were two patients here. The mother seemed to do okay. The baby seemed to do okay. And maybe the baby actually benefited from this by delivering the drug antenatally. So the take-home points, SMA is certainly treatable now. I think I hopefully convinced you of that. But is it curable? Well, we're not there yet. Maybe for those with 3 copies it is, but the 2 copies are still a challenge. Well, which drug to choose? We've got these 3 drugs. They all work really well. Well, that's for another day and time. Early diagnosis and treatment makes a difference. There's the opportunity for prenatal treatment, that we're seeing changed phenotypes in treated patients than the traditional type 1, 2, 3. That the traditional, what you read about type 1 and 2 will largely disappear. Newborn screening has had a huge impact. And there are other drugs in the pipeline. And importantly, standard of care is really important. You can't just treat them and treat them. You have to follow them in the clinic. You have to see, are they developing sensory or cognitive issues, and how are they doing? And is the effect in the gene therapy really sustained, or is it going to wane over time? So these are just the people involved. I think there's 62 people that I mobilized really quickly in 2 weeks to try to get this done. It was a huge effort, and I also want to acknowledge those at Roche and their U.S. Team at Genentech, and again, Annie Nguyen at the FDA. So it was a fairly big deal to try to get this done timely. And so these are other people who've just helped me over the years, including the hospitals I've been at and my funding supports and pharmaceutical opportunities, and people at the FDA who've actually, they're good people. They have to follow certain legislation, but they really are there to help, is what I found.
Wow, this is amazing. And several comments in the chat about how, for those of us in practice long enough to have had several patients pass away from SMA, this is especially incredible. So thank you for that. Can you tell us how you test motor function in the fetus? And then I'll ask it as a 2-parter. How do families choose between the different gene therapy options for SMA?
Right. So how do we test the fetus? We did what's called a biophysical profile which is an ultrasound assessment of the baby's kicking, breathing movements. It's the best we could come up with. And we also did what's called a fetal anatomical survey, which is a very detailed ultrasound to look at cardiac development, brain development, ventricular size, kidney. So the baby was getting weekly biophysical profiles, and then every 3 to 4 weeks, this anatomical survey. We didn't have any better way to look at that, but we're open to suggestions because that was obviously an important question. The second was how do we figure out which drug to recommend? Well, usually the parents come to us and say, I want the gene therapy. And you say, okay, that's fine, we can do that, but I just want to make sure you're aware that there are 2 other drugs. And what are the relative risks and benefits? Because they all work spectacularly well. And nusinersen, it's a bit of a pain because you got to do repeated lumbar punctures, but it's really, really safe, and it's been done in over 18,000 children and blah, blah, blah. And the onasemagene apavevac is still, we have 3,000 patients treated. There have been 2 deaths from the drug, from liver failure. It's pretty small, it's less than 1%, but it probably has a higher level of safety concern, as well as some other safety issues, thrombotic microangiopathy, other things that are rare, but collectively there's about a 1% risk of something pretty serious. And then the risdiplam also looks really, really safe, but compliance might be an issue because you got to remember to take it daily, like your whole life. So we balance that, but having said that, currently, I'm told, I don't know this for a fact, that 80% of parents are electing for the gene therapy if their baby's identified by newborn screening. The exceptions are if the baby is identified as having an elevated anti-AAV9 antibody, you're excluded from that treatment until the antibody level comes down, which could take a few months. So we urge them to start some treatment, any treatment, not just wait to do the gene therapy administration. So that means picking either nusinersen or risdiplam and then thinking about a switch over, which has its own issues of switching drugs. So, but that's the general scenario at the moment. Some cases it depends on the payer. So what their coverage policy is may dictate what the choices are, and that's particularly true in Europe. Other questions?
Thanks for such an outstanding talk. The success of gene therapy in SMA is highlighting, and you alluded to this, the need to confront the ethical issues about how to understand the long-term consequences of this treatment and equitable access to this very expensive treatment. Can you comment on how you see this success might apply, if it can be, to much more rare diseases, which you mentioned earlier in your talk, maybe outliers in the resources that pharmaceutical companies are willing to apply to them?
Yeah, that's a huge question. So one dimension of that question from Dr. Wainwright is this a sustainable model, this gene therapy? Because the drug is 2.125 million, the gene therapy drug. No, it's discounted a little, but okay, it's for you, 2 million. Okay. But okay, so think about 3,000 patients, $2 million each. Well, they didn't all pay it, but you get the idea. What about the next drug? Can the healthcare system absorb this? How are we going to pay for these? There are, I'm told, 450 gene therapy in preclinical development for different diseases. So let's say 10% of those work, because that's usually the case. There's a 90% failure rate for drug development. So let's say 10%. Well, can we absorb even another 40 at 2 million a pop? And there's some other gene therapies that are now 3 million. Okay, so there's this economic model. How are we going to do this? And can we do it? And then the selection criteria. Can we afford to treat everyone? So there's a whole new field of medical ethics that's evolving. And I've been privileged to participate in a panel, a group out of NYU Center for Bioethics that Art Kaplan leads. And there's a particularly thoughtful woman there, Alice in Bateman House, who's lovely and very thoughtful on this. And we're struggling with a lot of these topics, and there are no answers yet. So I didn't fully answer your question, but maybe just because we're at the hour, I'm going to stop unless we have time for anything more.
We're out of time, but thank you so much, Dr. Finkel, and all of your teammates for all of your great work in this field. And thank you to the Sarkowskys and Dr. Wainwright for inviting him. We really appreciate you. We'll be around in the room, but we have to end our recording. So thank you so much, everybody.
Yep. Thanks, everyone online. Okay.